Good morning, everybody. My name is Kendall.
And I'm Kelis.
How's your morning going so far?
My morning is going pretty well, actually. Yours?
It's going pretty smooth. Pretty smooth. What do you have to do today?
Well, all I have to do today is work, honestly. You?
I have nothing to do.
Free
So
day.
after this episode, I am completely free
Free.
for the rest of the day. Yeah, absolutely.
Well, let's get them introduced to what we're about to talk about so they're not lost.
Absolutely.
So, this is Let Me Sell You a Story, Season 2.
And on today's episode, we're going to talk about, hopefully, it's not too boring and we make it easy to kind of understand. We're going to talk about ribosome dysregulation and hepatitis C and how this virus affects protein production inside of the cell.
And we wanted this episode to feel more conversational and easier to understand because topics involving ribosomes and translation can sound really overwhelming at first.
Right. So instead of jumping straight into the scientific figures, we're going to break it down a little bit first. We're going to start with the basics, ribosomes, then go into hepatitis C, and then connect everything back to the actual research.
Right. So, first, let's talk about ribosomes because they're the main focus of our topic. Ribosomes are organelles responsible for making proteins through a process called translation.
And proteins are important because they control almost everything happening in the body, including structure, communication, enzymes, signaling, and chemical reactions.
Right. Ribosomes can either float freely in the cytoplasm or be attached to the rough ER.
And depending on where they're located, they make different types of proteins. So, free ribosomes are usually, those are the ones that make proteins inside of the cell, while ribosomes attached to the rough ER make proteins that are exported or inserted into the membrane.
So, basically, without ribosomes, cells wouldn't really function, right?
Exactly. If proteins aren't being made correctly, the whole cell can start having problems.
And normally, this process is very regulated because the body needs proteins to be made at the right time and in the right amounts.
Right, which is why viruses affecting this process can become dangerous. Would you say ribosomes are one of the most important organelles in the cell?
Honestly, I would say yeah, because almost everything depends on proteins being made correctly.
And I think people sometimes underestimate how important translation really is until something disrupts it.
Right. So, why does the cell need so many proteins in the first place?
Because proteins carry out most cellular functions. They help cells communicate, maintain structure, transport molecules, and even help speed up chemical reactions.
So, if translation is disrupted, multiple systems can be affected.
Exactly.
Okay.
Another thing that's interesting is that ribosomes are found in every living cell because every cell needs proteins.
And even though ribosomes are tiny, they have a huge impact on how the body functions overall.
Which is why studying ribosomes is so important in biology and in medicine. So, hepatitis C is a viral infection that mainly affects the liver.
And it spreads through infected blood, which can happen through shared needles, blood exposure, or contaminated medical equipment.
One thing that makes hepatitis C dangerous is that many people can have it for years and not even realize that they have it. So it's asymptomatic.
Yeah, so basically, symptoms can be mild at first, while damaging is still happening in the background without people knowing at all.
And over time, hepatitis C can lead to inflammation, fibrosis, cirrhosis, and even liver cancer without anyone noticing. So, why is hepatitis C considered dangerous compared to any other virus?
Because be- because it can become chronic and continue damaging liver cells over long periods of time.
And another important part of our topic is that hepatitis C can't survive on its own.
Yeah, viruses don't have their own ribosomes or machinery to make proteins.
So instead, hepatitis C relies on the host cell's ribosomes in order to survive and replicate.
So instead of the ribosome doing its normal job,
The virus can redirect it to make viral proteins instead.
so it's basically hijacking the system.
Exactly.
Okay. And I think one thing people forget is how important the liver is overall.
Exactly, because the liver helps with detoxification, metabolism, nutrient storage, and processing substances inside of the body. So many things.
So if the liver cells are constantly being damaged, multiple body systems can be affected.
And since hepatitis C can remain in the body for so long, that damage can continue over time. So, this is where the idea of ribosome dysregulation comes in.
Normally, ribosomes are controlled by the cell and make proteins the body actually needs.
But hepatitis C is able to redirect ribosomes to make viral proteins instead.
And one of the major ways it does this is through something called IRES, which stands for Internal Ribosome Entry Site.
Normally, translation requires multiple initiation steps and factors, but IRES allows the virus to bind directly to the ribosome.
So it basically shortcuts the process?
Exactly, which makes translation faster and more efficient, but for the virus.
Okay. And that's harmful because now the ribosome is focused on helping the virus instead of helping the cell.
Exactly. And over time, this contributes to dysregulation and cellular damage. And what I found interesting while researching this topic is how dependent hepatitis C is on host cell machinery.
Especially translation factors like EIF1A.
Right, exactly. The virus really depends on those host factors in order to function properly. That word always messes me up.
Always. So without those host translation factors, the virus would struggle to survive, basically, right?
Exactly. And that's why researchers are so interested in studying them. So, after understanding the overall concepts, we wanted to look at actual research to see how scientists study these interactions. So, this is where we bring in kind of like all our scientific information that we were able to research.
Right. And one thing we noticed across several sources that we were looking into was how dependent hepatitis C is on host translation factors.
Especially the EIF1A, which became a major focus in our primary article.
Right. So instead of only targeting the virus itself, researchers could potentially target how it uses the ribosomes.
Right, because if the virus can't properly use the host machinery, it becomes harder to replicate. So let's actually take a look at one of the figures we have here from the research that we were able to see. All right, I think you guys should be able to see it now.
So if you look at this figure, the researchers are trying to determine what proteins bind to the ribosome when hepatitis C is present. If you look at panel A, this is showing a pull down experiment.
And each band represents a protein. You can see factors like EIF2, EIF3, and EIF1A are all present.
So this tells us that the virus is recruiting host cell proteins to help translation occur.
And if you look over here at panel D, this is measuring how much binding is actually occurring.
And when mutations are introduced, binding decreases, which shows these interactions are important.
So, why is it important that these proteins are binding?
Because it shows that hepatitis C depends on host translation factors in order to function.
So, the virus isn't functioning independently.
Exactly. It's relying heavily on the host cell.
Let's take a look here at another figure we were able to look at. So, this one here focuses more on whether EIF1A is actually necessary for protein production. And if you look at panel A, I know there's multiple pictures in this, so luckily they're labeled, but if you look at A, this shows how they reduced EIF1A in the system. Now, if you look at panel B, they confirm that the EIF1A levels are very low. And then if you're able to focus in on E, G, and H, those panels are measuring protein production.
And what else we see is that when EIF1A is reduced, protein production also decreases.
Exactly.
So why did they reduce EIF1A?
To test whether it's actually important for translation.
And based on the results, it clearly affects protein production.
Exactly. This supports the idea that hepatitis C depends on EIF1A in order to efficiently make proteins.
So this isn't just theory anymore. This is actual experimental evidence.
Right. And that's why primary research is important because scientists are directly testing these ideas.
And I think this figure was one of the most important parts of the article overall.
I would agree as well because it directly shows how reducing the EIF1A affects translation.
Now we have another figure. So this figure summarizes everything together.
It shows how hepatitis C uses IRES to bind directly to the ribosome and recruit factors like EIF1A.
So this basically is like the bigger picture?
That's a perfect explanation of it. Exactly. It summarizes how hepatitis C manipulates ribosomes in order to make viral proteins.
And I think this figure helped make the overall mechanism easier to understand.
Definitely, because it visually connects everything we talked about throughout the podcast, and it brings the first figure, the second one, and the third one all together.
I agree. So let's come back to us. So before we wrap up, we want to answer a few listener questions related to today's topic.
Right. And hopefully this will help to bring it all together. So if anybody has any questions while we're answering them, continue to keep bringing them in, and then we'll keep going ahead and answering a few of those.
Right.
So I'll pull up the first one. It looks like Amanda from UNC asked, why does hepatitis C need ribosomes in order to survive?
Well, viruses don't have their own machinery to make proteins, so hepatitis C has to rely on the host cell's ribosomes to produce viral proteins and continue replicating. Another question we have is from Marcus from Georgia State. He asks, what makes IRES different from normal translation?
That's a good question, Marcus.
That's great.
Normally, translation follows a cap-dependent pathway, and it requires multiple initiation steps, but IRES allows hepatitis C to bind directly to the ribosome and bypass some of those normal steps. I think you went over that a little bit earlier today.
Yeah.
Let's see. Looks like Brianna from Howard asked, what role does EIF1A play in hepatitis C translation?
Well, based on the research we looked at, EIF1 helps hepatitis C efficiently produce proteins. Another question we have is from Daniel. He's from Duke, and he asks, why is understanding ribosome dysregulation important for medicine?
I can take that one because understanding how viruses interact with ribosomes can help researchers develop treatment that targets those interactions, and we need that research.
Right.
Let's see. Jordan from NC State asked, why does reducing EIF1A decrease protein production?
Because EIF1 is involved in translation initiation. So when its levels are reduced, translation becomes less efficient. Let's see. Well, we have a lot of people.
Keep them coming. Keep them
coming. I'm not. Hey,
we'll
let's
answer
bring
a
it.
few more. Yeah.
Right. Maya from Spellman asks, could targeting ribosomes help treat hepatitis C?
I can answer this one for Maya. Potentially, yes, because researchers are interested in targeting how viruses interact with host translation factors. I'll pull a question. Let's see. Chris from UNC asked, why do scientists study translation factors?
Oh, I got this. This is in the back. Okay. Because translation factors help regulate protein production. And many viruses depend on those factors to survive because they can't survive by themselves.
Exactly.
Let's see. Alexis from Howard asks, what was the biggest takeaway from this research? That's a great question.
That is a good question. Probably how dependent hepatitis C is on host cell machinery and how directly it manipulates normal ribosome function, I'd say. I'll pull a question here. Tyler from Wake Forest asks, can hepatitis C permanently damage the liver?
I personally would say yes. Over time, hepatitis C can lead to chronic inflammation, fibrosis, and even liver cancer if it's left untreated.
The cirrhosis as well.
Yes, that one too. Let's see. Amanda from Hampton University asks, why is protein production so important for the cells?
Good question. Let's see. Because proteins control most cellular functions, including structure, signaling, metabolism, transport, and chemical reactions.
Oh, yeah. I do remember you going over that. Let's see.
Let's see. I'll pull one. Kayla from Bennett College asked, what's the difference between a normal ribosome and a ribosome affected by hepatitis C? That is a really good question. That is a really good question.
Okay. Y'all listening.
Yeah.
Okay.
Keep the questions coming. I think we'll go over one or two more.
Yeah. Normally, ribosomes are making proteins that help the cell survive and function properly. They follow the cell's instructions and produce proteins the body actually needs. Compared to if it's affected with hepatitis C, it's not really focused on helping the cell. It's focused on helping the virus.
Absolutely. When hepatitis C affects the cell, the virus basically redirects the ribosomes to focus on making those viral proteins instead.
Right. Let's see.
I think also, and that's where dysregulation happens because the ribosome is no longer fully working for the cell. It's also helping the virus replicate.
Right. And that's where dysregulation happens because not the protein, the ribosome is no longer fully working for the cell, but it also helps the virus replicate as well.
Absolutely. Let's see here. You want to ask the next question?
Oh, yeah.
Let me see.
We
All right,
got
then.
Michael. You
Yeah.
can do Michael. Okay. Michael from UNC Charlotte asks, why can hepatitis C use ribosomes so effectively?
Yeah, good question. One major reason is because of IRES medicated translation. So normally the ribosome has to go through several regulated factors, initiation steps before translation even begins.
Right. But hepatitis C uses IRES to bypass some of those normal control mechanisms and bind more directly to the ribosome.
Which makes the process more efficient, I would say, for the virus because it doesn't have to rely on all the same normal cellular signals.
Right. And the virus also recruits host translation factors like EIF1 to help stabilize and support translation.
So, for that question, overall, hepatitis C is very effective at taking advantage of the host cell's machinery.
Right. Let's see. We got one more question.
Yeah, let's go ahead and answer one more question. It looks like it's from Jasmine from A&T. She asks, why is this research important outside of just hepatitis C?
Well, I think it is important because it helps scientists understand how viruses interact with cells in general, and research helps scientists develop antiviral treatments, just in case.
Yeah, absolutely. And research is important just outside of, like she said, hepatitis C. We need research in order for medications to happen for multiple reasons so that we can understand it as well.
Right. Well, just to summarize everything we talked about today, we started by discussing ribosomes and their normal role in the cell. Ribosomes are responsible for making proteins through translation, and proteins are important because they control so many functions in the body. We also talked about how ribosomes can either float freely in the cytoplasm or attach to the rough ER, depending on what type of proteins they're making. And normally, this whole process is very regulated because the body needs proteins to be made correctly in order for cells to function properly. Then we connected hepatitis C and explained how it's viruses that mainly affects the liver and depends completely on the host cell in order to survive. Since viruses don't have their own ribosomes, hepatitis C has to use the host cell's machinery to make viral proteins and continue replicating. We also talked about how hepatitis C can become chronic and lead to long-term liver damage, which is why understanding how it interacts with the cells is so important. So overall, this topic really showed how something so normally helps the body function, like ribosomes, can actually be manipulated by a virus without us even knowing.
Yeah, absolutely. And just to add on a little bit to that, we also focused a lot on the actual mechanism behind hepatitis C affects translation and ribosome function. One of the main concepts we talked about today was the IRES, which allows the virus to bind directly to the ribosome and bypass some of the normal translation steps. And that's important because it helps the virus take control of protein production more efficiently. Instead of the ribosome focusing on the normal needs of the cell, it starts producing viral proteins that help hepatitis C survive and replicate. We also connected this to actual research from our primary articles, where you did see those figures come in figure one, we saw that translation factors like EIF1A were able to bind during hepatitis C translation, which showed that the virus depends on host cell proteins. Then in figure two, we saw that when EIF1A was reduced, protein production was decreased, which supported the idea that this factor is important for translation. And then finally, in figure six, it helped us summarize the entire mechanism and showed how hepatitis C uses IRES and translation factors to interact with the ribosome. So, overall, this project really helped us understand how viruses can manipulate normal cellular processes and how research can be used to study those interactions in more details.
Right. So today we talked about ribosomes, hepatitis C, and how viruses can dysregulate normal cellular processes.
And how research can help us better understand those mechanisms at the molecular level. So hopefully it wasn't too boring. We were able to break down ribosomes, break down hepatitis C, how those ribosomes are dysregulated when it does have hepatitis C, and just give us some of that back information, which is the scientific figures that we were able to pull in from our primary article. So hopefully we didn't make it too boring for everybody, and you leave the podcast today learning something.
Right. I hope we answered all y'all questions correctly. Sorry for us laughing. We tried.
We had some really good questions come over from all over, which was really, really good. So that was awesome. You guys can keep sending in questions, but I hope, again, that you learned something from today's podcast.
All right.
Thank you guys so much.
Thank you.
And we'll see you next time.
Goodbye. Enjoy your day.